Okay. Okay, guys, let's try to move on to the next one. Okay, so next question I want answer from Darshanita especially. Okay, so Darshanita, tell me. Okay, recombination between homologous chromosomes is completed by the end of what? Yes, very nice, very nice. Yes, everybody. Rest others also you try. Yes, answer is patching. Very nice. Okay, it is going to be patching. Okay, next question is for Divya Savant. Divya, crossing over takes place in. Divya, quickly, quickly, quickly. Divya, are you there with us? Divya Savant. Rest others you can answer quickly. Yes, Divya Savant is nowhere to be found. Okay, so we move on to Zoe. Zoe has already answered. Yes, what is it? The answer is prophase 1. Right. Okay, next we are going to move on to Zoe once again. Zoe, homologous chromosomes separate during. So we are talking about separation of homologous chromosomes. Very nice, Zoe. Very nice. Danishka has also given me the right answer. Yes. What is it? It is going to be during your anaphase 1. Right. Okay. Now, next thing, microtubules from the opposite poles of the spindle get attached to the kinetochore of the chromatid of sister chromatids during. Yes. Guys, tell me. Microtubules from the opposite poles of spindle fiber get attached to the kinetochores of the sister chromatids during what? No, 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 no. Zui has given me the right answer. No, 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 no. Rest others are not giving me the right answers. Guys, attachment of spindle fibers to your chromosome. It is a characteristic of what phase, guys? It is a characteristic of prophase. So it is going to happen either in prophase or in prophase 1 or in prophase 2. Now here, guys, ideally it should have been prophase 1, but then there is no option of prophase 1. So what are you going to go for? You are going to go for prophase 2. Guys, get this thing. This is a trick question. Okay, they want you to feel that अरे नहीं ये पहला meiosis 1 में से है तो we'll go for meiosis 1. Okay, in order, but no, it is not that way. We have to see whether it is prophase or no. Okay, next question, Kanaka, I want answer from you. Exchange of segments between the non-sister chromatids of homologous chromosomes. Exchange. Yes, 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 yes. It is going to be crossing over. Very nice. Okay. Next question is for Rishik. Rishik, in meiosis, synapses occurs during. Yes, very nice, very nice. But waiting for Rishik. Rishik, quickly, quickly, quickly. Rishik. Seems like Rishik is not there, but then yes, others have answered. So yes, it is going to be your prophase. And guys, quickly, just quickly chat and tell me in prophase which phase are we talking about? For synapses, we have very nice. It is zygotine. Very nice. Yes. Okay, chalo, next one. Okay, now this question is very important, guys. I want you to understand this thing. And Ruby, you are going to answer this especially. Meiosis involves two nuclear division, one chromosomal division, one nuclear division, and one chromosomal division, one nuclear division, two chromosomal division, and two chromosomal divisions and two nuclear divisions. What do you think? Very nice. Very nice. Guys, think about this thing. This is a very important question, actually. Think about this once again. Guys, just take a moment. Okay, so sorry. Say, guys, when we talk about your this thing, meiosis, okay. So some of you, sir, in the previous question, prophase one should have been mentioned. Yes, actually, it should have been prophase one. That is true. It should have been prophase one, but then, because we know, we understand that it is that way. That is why we have selected this, and this is the best answer from this. Okay, so now, guys, we need to understand out over here. Okay, meiosis involves what? Meiosis involves. One minute. Just a second, guys. Yes. Okay. Okay. So, guys, once again, see what is going to happen. Most of you have said A, some of you have said B, some of you have said C. Now, guys, listen. What is acting out over here? See, guys, meiosis is going to have two nuclear divisions. What are those two nuclear divisions, guys? Karyokinesis one, and it is karyokinesis two. Aren't these nuclear divisions? Yes. So nuclear divisions, they are going to be two. But one chromosomal division. Yes, one chromosomal division where? Guys, this is going to happen in your anaphase 1, because the chromosomes are going to be divided equally. I mean, it is going to be divided, it is going to be reduced to half. So yes, this is going to be only once. The same thing is not going to happen in your anaphase 2, right? So guys, it is going to include two nuclear divisions, and it is going to have one chromosomal division. Okay, so guys, remember this thing. This is a very, very important question. Okay, this is a trick question. So answer is A. Very, very important. Okay, next thing. Okay, in which stage of meiosis the structure, number, and shape of the chromosome is best observed? Yes, it is going to be yes. Everybody without a hair, oh, there is still a wrong answer. But then what is this? It is going to be metaphase one. Guys, no matter whatever it is, whether it is metaphase one, whether it is metaphase two, whether it is just a metaphase in mitosis, chromosomes are going to be best sent out over there. Okay. Next, in which stage synaptonemal complex dissolves, chromatids become clear, and bivalents are called tetrads? This is very important, guys. Okay. So, what are they saying? The synaptonemal complex dissolved. Now, what was the synaptonemal complex, guys? The synaptonemal complex is nothing but your synapse. Okay, when is this thing going to dissolve? The chromatids become clear and bivalent, meaning the chromatids are going to become like this. They become like really, really clear, and they are going to be now known as your tetrads. Yes, guys, can anybody think of this thing? Okay, please try this, because this is also very important question, guys. Very important question. Okay. Yes, 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 yes. Some of you are right. Some of you are still wrong, but still, once again, okay. And plus, you are going to see that this thing becomes really, really like it becomes like really, really proper. And when they are going to look the best, that is what they are trying to see. Okay, guys, the answer for this thing is diplotin. Okay, once again, guys, this is very, very, very important. You will be always tempted to say pachitin because in pachitin, these things become like, really, really thick. But no, guys, the answer is diplotin. See, what are they saying? The synaptonemal complex is going to dissolve and chromatids become very, very clear. Now, why it is going to happen that way? See, the first thing is dissolution. Okay, tetrads, we are anyways going to call them after crossing over, we can call them as tetrads or bivalents. That is fine. But the dissolution of synaptonemal complex, this is going to happen in your diplotin. So once again, guys, what exactly is happening? You are going to see that, oh, both of these, they are going to be like separating apart from one another like this. They are going to move away from one another, and that is the time when these chromatids, they look the best, they are going to appear the best. And that is why, guys, the answer is diplotin. What is the synaptonemal complex? It is the synapses basically. The synapse, the synapses or the synaptonemal complex. This is synapses. Okay, sure. Let's try to move down. Okay, everybody has got this one, right? Guys, these are like a little trick questions, okay? Where you'll be tempted to answer something else only. Okay, sure. Guys, next one, the answer is for Tanishka, okay? Tanishka, at what stage does the chromosome number become half? Very, very simple and straightforward. Yes, very nice, very nice. It is going to be anaphase 1. Okay, next one is Vaishnavi. Vaishnavi, when synapsis is complete all along the chromosomes, the cell is said to have entered into a stage called dash. When synapsis is complete, okay? Yes, think about it, guys. Yes, very nice. So once this is complete, guys, this is now going to enter into your patching. Okay, because after your zygote, we are going to have patching. Okay, next question is for Akanksha. Akanksha, meiotic cell division is also termed as reductional division because very simple, guys. Everybody, quickly. Yes, yes, yes. Very nice. No, 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 no. Why, guys? Why are we calling this reductional division? Because the chromosome number is half. That is why. Okay. Next thing, chromosome number. Ankita. Ankita, chromosome number is restored by. Yes, it is going to be your meiosis. Some people are still saying B. Come on, guys. This is A. Chromosome number is restored only by meiosis because first there is reduction and then there is this thing. Then there is fertilization and I mean, see, once again, guys, see, we were 46, 46. By meiosis, you said you are going to get 23 in the sperms and 23 in the ovum. And when this thing is going to happen, guys, once again, you are going to get 46 in the zygote. So it is because of crossing over and not mitosis. I don't know. Some people are saying mitosis. Why I don't understand. Okay, next thing. Okay, in meiosis, centromere divides during. Okay, guys, once again, very important. Yes, yes, very nice, everyone. Answer is anaphase 2. What was this we are talking about, guys? See, this was the X-shaped chromosome, and we said, oh, the X-shaped chromosome is going to divide from within the center. In your anaphase 1, what had split, guys? Can anybody quickly tell me? Anaphase 1, what did split? Chiasma. Very nice, Kanaka. Yes, the chiasma had split. Okay. In anaphase 2, your centromere splits. Okay. Next thing, variations appear during meiosis due to, yes. Now I want Ankita to answer this thing. Ankita, can you tell me variations appear during meiosis due to? Okay. Now, guys, the real answer for this is both independent assortment and crossing over. Okay. So, what is this independent assortment and what is this crossing? अरे रे ये क्या हो गया? One minute. Huh. So what is this independent assortment? Guys, independent assortment means it is like, whatever chromosomes that you have, those chromosomes are going to be free. They are free to move anywhere. So they are not going to be like, oh, I'm not going to like, mix with this. I'm not going to mix with that. No, here you are going to see that every gamete, which is every allele which is present, it is capable of like mixing with any other allele, and that is nothing but your independent assortment. So if you just, I will have to, if I have to talk about it, you know it is actually your Mendel's third law. Okay, and what does the law states? Okay, the distributions of the members of the allelic pair into the gametes is independent of the other allelic pair. So meaning what, guys? To be in very specific thing, everybody is free to mix and match with everybody else, so that you can get new combinations, and that is nothing but independent assortment. So guys, here, yes, it is both independent assortment also and crossing over. Okay, next question is for Arshia. Arshia, quickly tell me the stages, proper sequence of prophase 1. Yes, very nice, very nice. It is going to be leptotene, zygotine, pachytene, diplotin, and diakinases. Yes. So, guys, once again, right now, someone had asked me recombination nodules. Now, where are you going to see recombination nodules, guys? Yes. Quickly. Just think about this, guys. Yes. It is. See, now there are anaphase, metaphase, prophase, telophase. So, definitely it has to be your prophase, because in prophase you are going to see this thing happening. Okay. Okay, and generally, where do these recombination nodules start? Okay, remember this thing, recombination nodules. Okay, they are going to start during your zygote. Okay, because at that time, we are going to see synapses and all starting to take place. Okay, so that way. Okay, it starts being like we see this thing. Okay, at the time of your zygote. Okay, it starts from there. Meaning. Okay, but answer is prophase. Okay, next question. Which one ensures maintenance of chromosome number generation after generation? Yes, guys. What do you think? Very nice, guys. Very nice. It is going to be meiosis only. I right now explained at the top, okay, because the chromosome number is going to be reduced to half, and at the time of your reproduction, I mean, at the time of your fertilization, it is going to be brought back to normal. Okay. Next question, Darshanita. Terminalization occurs during. Quickly. Yes. Very nice, everyone. Yes. Darshanita hasn't answered though. Darshanita, quickly. Are you there with us? Darshanita. Yes. Okay, what is it? It is diakinases. Yes. Okay, next question for Divya Sawant. Divya, tetrad is made up of, guys, very important question. Okay, this is very important. Okay, you have to understand what is happening. Yes, so I want answer from Divya Sawant, but then rest others quickly tell me. What is a tetrad? No. No. Yes. Darshanita is right. Vaishnavi is right. Kanaka is right. Yes. Zui. Zui has not answered. Yes. Zui is also right. Yes. So, guys, what is the answer? The answer. See, what is a tetrad, guys? A tetrad is this thing. Okay, we had said. What do you mean by tetrad? We have said there should be four chromatids. That means it is a tetrad. Okay, so how many homologous chromosomes, guys? These were homologous chromosomes. These are two homologous chromosomes. Two homologous chromosomes, and how many chromatids, sister chromatids? Four sister chromatids. So, four, out of that, there are two sister chromatids, and these two are two sister chromatids. So, yes, it is two homologous chromosomes, each with two chromatids. Okay, guys, understood this thing? This is very, very important question. Okay, these kinds of questions they will always ask. Like, they may ask you sometimes. There is 20 tetrad formation in a cell. A scientist find out that there were 20 tetrad formation. Find out this thing. Find out how many chromatids will be seen that way. Okay, so you need to understand what is a monad, what is a dyad, and what is a tetrad, guys. Okay. Next one, I want answer from Zui. Zui, meiosis 2 performs. Meiosis 2. Very nice. Very nice. Everybody is giving me the right answer. It is separation of chromatids. Guys, remember, separation of sex chromosome. This happens in anaphase 1. Okay, don't mark this thing. Separation of chromatids. This is going to be in your anaphase 2. Okay, chalo. Everybody getting this? Fine. Chalo. Next question. I want Kanaka to answer. Kanaka, poleward movement of dyads occurs during. Very simple. Now we are talking about dyads. Okay, we are not talking about tetrads. Yes. When is it? Yes, it is going to be during your anaphase 1. Very nice, guys. Very nice. Okay. Next question is for uh, Kanaka. Kanaka, DNA chromosome replication or chromatid formation takes place during. Very simple. Okay, guys, I have always told you when we are going to talk about the DNA, whether we are talking about the duplication of chromosomes, whether you are talking about the increase in the nuclear content, whether it is your histone protein formation, whatever that is going to happen with your DNA, guys, which stage it is going to be your S phase. Okay, everybody getting this thing? Sure, no problem. Okay, let me just. Okay, so this was one. Okay, now I'll just we'll move on to the other one, guys. Okay, sure. So let's try to see the next one. Okay, is everybody able to see this thing, guys, properly? Is it blurred or is it okay? Yes. Okay. Someone is asking dyad means two chromatids. Yes, dyad means two chromatids. See, once again, guys, anybody who has having confusion, let me just once again put it down. See, this is monad. Okay, we had said this is monad chromosome. This is a single chromosome. Actual chromosomes are these. What is a dyad one? Dyad are these. And then we are going to have tetrads. Tetrads are these. Okay, you need to know the difference between all the three, guys. Okay, very, very important, because in your exams they are going to quiz you on this thing only. Okay. ये क्या है? Okay, chalo. So guys, once again, let's move ahead. I want answer from Rishik out over here. Rishik, in which stage chromosomes have reached their respective poles, decondense and lose their individuality? So tell me. Very nice, very nice. Those who have messaged, very nice. Yes, guys, what are we talking about? We are talking about, yes, it is going to be your telophase, guys. Yes, it's going to be telophase. Okay, chal. So guys, next one. Let's try to move on to the next one. We have, okay, telophase is characterized by. Okay, the answer I want answer from Ruby. Ruby, telophase is characterized by. Yes, it is going to be all of the above. Very nice. Okay, read the following statement and find out the incorrect statement. Okay, so the first one I want answer from Tanishka. Tanishka, uh, uh, Tanishka, do you have a brother by any chance? Okay, 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 okay. All right. Chalo, Tanishka, tell me. Read the following statements and find out the incorrect statement from the following. Okay, we'll do this step by step, one by one. In an animal cell, cytokinesis is achieved by the appearance of furrow in the plasma membrane. What do you think? Is this right or is this wrong? Tanishka. This is wrong. Okay. Tanishka is saying this is wrong. So, Tanishka, can you tell me then if it is wrong, then what is the correction out over here? Tanishka, can you be a little quick, please? Rest others, kindly tell me what do you think about this statement? Is it right? Is it wrong? I have got answers from Zui and Kanaka, but others also want to participate in this thing. Yes, guys, this statement is actually right. This is not incorrect. This is actually right. Okay. Okay. So this is right. Okay. Let's try to move on to the second statement. Okay. Now, Tanishka, once again, try to tell me. Okay. Furrow formation starts. Okay. Actually, why I asked, Tanishka, because, I had a kid whose name was Anish Angane. So I thought maybe you're his sister. Anyways, okay, furrow formation starts. Acha, oh, okay, okay, okay. Okay, furrow formation starts in the center of the cell and grows towards the out to meet the existing lateral cell walls. Oh, is it true or false? This is the wrong one, guys. This is the wrong one. Let's try to see if the other statements, are right or wrong. Cell plate represents the middle lamella between the walls of two adjacent plates of plant cells. What do you think? Yes, this is right because we are going to have this middle lamella out over there. We have primary cell wall, then we are going to have secondary cell wall. Yes, that is right. And let's see this one. During cytokinesis, organelles like mitochondria and plastids get distributed between the two daughter cells. Is it true? Yes, this is also true. And here, guys, there is no like division, like equal division of this thing. Okay, they are going to be just divided randomly. Cell organelles are divided randomly. Okay, so understand this thing. So, who is the wrong statement? This is the wrong statement, guys. Okay, next case, next this thing, Vaishnavi. Multinucleated condition arises due to karyokinesis is not followed by cytokinesis. This is called as what? Yes, this is very important, guys. This question is asked always. And those who have been with me in ninth standard, okay, guys, you will remember we have done something of this thing in economic importance of your fungi. Yes. What is this? This is nothing but we call this thing as syncytium. Okay. So remember, guys, this is something syncytium. And where do we generally see this syncytium, guys? This is generally seen in your fungi, in your cenocytic hyphae. So generally the fungi, it is going to show you something like this. And these are the nuclei out over here. Okay, but then what happens, guys? You are going to see that now these lateral walls. अरे ये क्या हो गया? Okay, जाने दो इधर थोड़ा सा. This is a little different than what I normally use. Okay, so now what are you going to see, guys? You are going to see that this is going to change into this, where you're going to see only this thing, this lateral walls, these walls, they are going to dissolve, and you are going to see that, oh, this is going to be multinucleated without any kind of this thing. This is nothing but your syncytium, and such kind of a cell, it is called as a syncytial cell. Okay, fine with this thing. Okay. Now let's try to see this. Oh, now this either many answer. I half of the answer I've given it out over here. Okay, but now, guys, syncytium is seen in. Now, where are we going to see this thing? So, guys, think about this thing. Yes, yes, it is going to be your okay. There are like phycomycetes, ascomycetes. Okay, what are all these? Let's try to see. Okay. Then we also have like something that is endosperm of the coconut. Okay. And mammalian RBCs. Mhm. Okay. Okay, chalo. So guys, let's try to see this thing. Now I have told you, yes, this is going to happen in your fungi. Okay, but now which class of fungi? That is also very important. Remember, all your phyco this thing. Okay, all of them, they are not going to show you this kind of sensation formation. Phycomycetes, they may not show you. Ascomycetes, that means your back fungi, they may also not show you. Okay, then there is basidiomycetes, club fungi. Okay, so the chances are that here we don't know what they're really talking about, but guys, here there is one example which 101% shows sensation. And who is that? That is nothing but, guys, it is going to be your liquid endosperm of the coconut thing. Okay, guys, so when we have that coconut water, okay, what is this coconut water? Coconut water is nothing but the endosperm of that coconut seed. Okay, and now what is going to happen, guys? Inside that liquid, basically the cells are just dividing without proper cytokinesis. That is what is going to happen, guys, and that is why we'll see that you are going to encounter this in the endosperm of your coconut. Okay, very important question, guys. Just keep this thing in mind. Yes, I'll send these files today. Okay, so guys, keep this thing in mind. Okay, sometimes you may be tempted to you know say fungi. Okay, but then answer, just look out for this answer. This is a well-known typical answer, guys. Okay, next question I wanted from Akanksha Mulik. Akanksha, what is the significance of mitosis? By the way, Akanksha, how's your sister doing? She's fine? Okay, and what is she doing now? And probably second or third year, right? Second. Okay. Acha, chalo. Akanksha, tell me, what is the significance of mitosis? Very simple. Yes. Answer is all of the above without even thinking, guys. Okay. Next question. Okay. Next question is for Ankita. Ankita, a very significant contribution of mitosis is cell repair. Repairing takes place in our body. In. Yes, it is going to be all of the above. Very nice. Very nice, guys. Very nice. Okay. Next question is for Arshia. Arshia, in which of the following stages cell division chromosomes are most condensed? Very simple, guys, very simple. Yes, it is going to be your metaphase, right? Very nice. Okay, next one I want Darshanita. Okay, shape of the chromosome can be best observed. Very simple. Once again, guys, yes, it is going to be your metaphase. Same thing. Okay, next I want Zoe. Major events that occur during anaphase of mitosis, which brings about equal distribution of chromosomes, is yes. Others also answered no, 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 no. Read the options properly, guys. There are two options which may seem to be right, but out of that, only one is technically right. Very nice. Very nice. Okay, guys, what is the answer? See, there are two options I said which are close. One is splitting of centromere, and second is splitting of chromatid. But guys, what is the right answer? Splitting of centromeres, guys. Because if I say there is splitting of chromatids, who knows how the chromatids are splitting? Are they splitting like this? We don't know. They are not talking about anything out over here. So this statement actually itself in itself is wrong, guys. Okay, because splitting of chromatid is not right. Okay, splitting of chromatids is wrong. It is splitting of your centromeres, and that is why you're going to see separation of sister chromatids. Okay, chalo, let's try to move ahead. Next one. Okay, I want Kanaka to answer. Kanaka, chromosome are least condensed in. Yes, 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 yes. It is going to be your interface. Very nice. Yes, it is interface. Okay, next question is for Rishik. Rishik, centromere is required for. Yes, very nice. It is movement of chromosome towards the pole. Okay, actually, these are just a random things that they have given. Okay, there are more important functions of centromere as well, but then still, this is the best one from the out of the given options. Okay, Ruby, which of the phases of mitosis is the longest? Yes, very nice, very nice. It is prophase. Okay, in mitosis, centromere divides. Rishik? Yes, very nice. It is going to be in anaphase. Okay, next question is for Tanishka. In plant cell, cytokinesis occurs by cell plate, invagination, furrowing, all of the above. What do you think? Yes, very good, guys. Very good. This is cell plate. But guys, then what do we mean by invagination? So actually, when the cell membrane it is like going in like this, that thing is called as invagination. Invagination meaning falling down or folding of your cell membrane, or it can be nuclear membrane. That is called as invagination. Okay, so this thing is invagination or furrow formation. Okay. Next question. Cytokinesis, Akanksha. No, Vaishnavi, Vaishnavi. Vaishnavi, yes. It is very nice. Division of cytoplasm. Okay. Next question. Okay, Arshia. What is the proper sequence in mitosis? I told you, without a hitch, you should give me the right answer. Yes. Very nice. It is PMAT. Prophase, metaphase, anaphase, telophase. Darshanita. Prophase 1 is subdivided into 5 phases based on. Yes, it is going to be, guys. I want every one of you to answer this thing. Prophase 1 is divided into 5 phases based on. Think. Look at the answers, guys. Look at the answers. Yes. Think about it. Think about it. Only one person has given me actual right answer. Okay, guys, what is it? It is your chromosome behavior. What exactly was happening? See, earlier the chromosome was like this. In the next case, the chromosomes become like this. Leptotene, it became like this. Zygotine, now they are like this. In pachytene, now they are going to be like this. Again, diplotin, now they are like, moving away. So this is all behavior. Okay, guys, understand this thing. This is behavior, chromosomal behavior. Okay, most of you have got this thing wrong, so keep this thing in mind. Okay, now a shape of the chiasma is, yes, what do you think, guys? Yes, it is always going to be X-shaped chiasma. Okay, next question is chalo for Arshia. The beginning of diplotin is recognized by something very, very important. We just now said. Okay, so let's try to see. Yes. No. See. Some of you are giving me right answer, some of you are giving me wrong answer, but let's try to see. The beginning of diplotin is recognized by yes dissolution of synaptonemal complex. That is what we did in the earlier this thing also. Okay, in the earlier question. Okay, now tendency of the recombined homologous chromosomes. Okay, tendency of the homologous chromosomes of the bivalents to separate from each other. So is this really right? Like, are they like, the tendency of this thing? This is a little off. Okay, mitotic spindle is assembled. That is not this thing actually. So answer is, out of these two, the best answer would be A. Okay, rather than going for D, A should be the best answer, guys. Oh, 1 minute. Sorry. Let me just see. Tendency of the recombined homologous chromosomes. No, answers will be both A and B. Okay, both of them are going to be right, because here they are just separate. They are just tendency of separating. So they are not separated completely. They are just saying that, oh, they are just trying to separate from one another. I did not read this thing properly. Answer is actually both A and B, guys. Okay, I did not read the complete statement properly. Okay. So next one, the stages through which a cell passes from one division to the next division. It is called as, yes, guys. No. Just think about this thing. Wait. The stages through which a cell passes from one division to the next division is called. It is nothing but the sequence of events associated with the life of a dividing cell. Yes, what is this? This is nothing but cell cycle. Okay, everybody getting this thing? Fine? Okay, just a second. I will just save this thing. Okay, I hope everybody is getting the hang of this thing. Like, everybody is understanding what is happening. Okay, on similar lines, guys, next class when we come back now, I will once again, scan that book and I will like upload it out over here. I don't know what happened, where those scans went. So that one I will upload, and then we'll do that thing. That is like, that is like, how it will actually come in your exams. It is that way. Okay, I still have one more, but I'll just show you the other one first. And always remember, guys, what is the key for successful this thing, for the clearing of need? More and more practice. The more you practice, guys, the more simple it is going to be. Okay, and bio, I'm I'm telling you guys, if you stick with me for the next 2 years, guys, out of 360, 340 question, I mean, marks worth 340, you will definitely know. Only for the next 20 marks, that is only for 5 questions, you will have to really read. Rest, everything will be covered in this entire this thing, guys. Okay, chalo. So guys, let's try to see this thing. Okay, what is the average life cycle of your yeast cell? Yes, can anybody tell me? Anybody, does anybody know? Yes, very nice, very nice. It is going to be 90 minutes. Yes, good, 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 good. Sorry, what is happening? 1 minute. Just a moment. Sorry. No, it is not allowing me to use my pen only. Huh, just a second, guys. Wait, let me just restart this thing. Okay, chalo. I don't know what is happening, but once again, it is not allowing me only. Okay, okay, let's see this thing. Okay, okay, so I will just. Anyways, this one I'm not able to edit this thing. I don't know why. What is wrong with this? Okay. Anyways, chalo. First thing is, yeah, actually I do that, but then it would have eaten up a little time now. So that is why I was like, chalo, isme we'll do it. Okay, chalo. The first answer is right, guys. First answer is 90 minutes. Let's try to move ahead. The life cycle of a cell is divided into interphase and cell division stage. The interphase is subdivided into substages. The interval between mitosis and synthetic phase is referred to as. Guys, very nice. Very nice. Guys, read the option properly. Guys, the see. Okay, the interval between mitosis and synthetic phase. Okay, so we are going from mitosis and we are entering into your synthetic phase, meaning the in between that it is going to be your G1 phase. Okay, remember, very very important. Otherwise, you know what you may think, sir, but G2 phase is also right, because वो भी तो उल्टा इसमें आता है synthetic phase and mitosis. But then if that they were talking about G2 phase, rather than saying mitosis and synthetic phase, they would have said synthetic phase and mitosis. Okay, so get that thing. Okay, next one. We are having in synthetic phase of the cell cycle, which of the following structures get duplicated? Yes, what do you think? Yes, it is going to be your DNA. Very nice, everybody. Very nice. It is DNA. Okay, guys, meiosis 2 performs. Yes, what do you think? Repeat question. It is separation of chromatids. Yes, very nice. Okay, now guys, this is a little important question. Okay, Balbiani rings. Okay, and this is like, keep it in mind. Okay, always keep this thing in mind. Balbiani rings are sites of. Now, on your DNA guides, you will see like, there are small, at some case, in some cases in your this thing, you will see in prophase one, especially, you will see like, suddenly you will see like small, like, bulbs, bulby and kind of, appearance of your DNA. Why it is happening is, it is because of RNA and protein synthesis at that point. That is why it is happening. Okay, so guys, keep this thing in mind. Actually, okay, this is a very important question. Balbiani rings, they appear in your prophase one, and why is it? It is to show that RNA and protein synthesis is taking place. Okay. Okay, next one. Okay, guys, this one is also very important. Okay, number of chromatids at metaphase is, okay, just you have to compare this thing, guys. Yes, okay, very nice, very nice. Tanishka has given me the right answer. Tanishka is actually right, Zui is right, Darshanita is right, Rishik is also right. Yes, yes, yes. Okay, so very nice, guys. Okay, so guys, what is the answer? Arey, re. Just a second, guys. Just a second. What I will do is Control All, Control C, Control New, Control V. Just a second, guys. Okay, chalo. So, guys, once again, we are in OneNote. Okay, so let me just zoom. अरे ये क्या हो गया? Okay, just let me know, guys. You can see this thing properly, right? Everybody? Yes. Okay, chalo. Take it. Okay. So once again, this was. अरे यार, I don't know what is happening. Okay. Okay. So first one was 90 minutes. 90 minutes. This was your G1 phase. Then this is your DNA. Then meiosis 2 helps in the separation of chromatids, Balbiani rings, RNA, and protein synthesis. Okay. Okay. So number of chromatids. Yes, this is going to be now. How do we do this thing, guys? Okay, let's understand this. Okay, so always remember when we are talking about 1 minute. So when we are talking about your chromosomes, okay, so let's imagine we are talking about your metaphase. Okay, in metaphase of your mitosis, you're going to see a situation like this. And in your metaphase one of your meiosis, you're going to see a situation like this. So see here, there are two chromatids, but here there are going to be four chromatids. If you just compare this, so two and four, so you will see two in mitosis and four in your meiosis. Okay, everybody has understood this thing, guys? Okay, fine. Hold 1 minute. Okay, guys, next thing. During your mitotic anaphase, chromatids migrate. Yes. Chalo, I want Vaishnavi to answer this thing. During mitotic anaphase, chromatids migrate. Yes, 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 guys. Very nice, very nice. Vaishnavi has given the right answer. Waiting for the others. Zui is the right. Darshanita is right. Kanaka is right. Yes. Where is it? It is going to be from your metaphase towards the poles. Very nice. Okay. Chalo. Next one, Akanksha. I want you to answer this thing. Okay. So microtubules that form spindle. Okay. Microtubules that form spindle tend to originate from dash and terminate into dash. Very nice, very nice, very nice, guys. Very nice. Zui is right. Darshanita is right. Guys, waiting for others. Rishik, you have become a little passive. You are not answering. Rishik, Tanishka, try answering, guys. Yes, what is it? It is going to be centrioles and kinetochores. Very nice. Okay, synaptonemal complex is more conspicuous at when it is going to be like more conspicuous. Yes, guys. Quickly. Yes, okay. Now, guys, once again, see, there is a little thing, okay, which you need to think about once again, okay. So, guys, when is it going to be formed? It is going to be formed in your zygotine. Okay, but in zygotine, what is happening is in zygotine, it is not going to be that prominent because the chromosomes and all they are not going to be looking that great. But now, when it is going to look the best, okay, it now always remember synaptonemal complex. It starts in your zygotine. Okay, but when does it look the best? It looks the best in pachytene. Guys, this is a catch. Okay, so you need to keep this thing in mind. If formation is in zygotine but looks better in your pachytene because at that time it is condensing and the chromosomes become like really nice. Okay, next thing, in a plant cell. Okay, guys, let's try to do this thing. In a plant cell, cytokinesis is accomplished by the formation of a very simple. Yes, it is going to be cell plate. Okay, next question I want Rishik to answer. Rishik, the distribution of mitochondria and plastid between the daughter cells during cytokinesis. I told you a few moments ago. Yes. Very nice. It is going to be random. Okay, when the dividing cells are examined under light microscope, chromosomes first become visible during. Yes, what do you think? Chromosomes are going to become visible during. Yes, it is going to be during your prophase only. Okay, next question. Centrosomes. Okay, my question goes to Darshanita. Centrosomes are dash. Yes, guys, read the options properly. Centrosomes are dash. No. Uh-huh. These are nothing but really constricted regions of your chromosome. So we had said, see, that is like, constriction prime. So actually the other name for your sorry, the guys, no, this is centrosome. They are not asking centromere. Guys, read this question properly. Okay, this is a centrosome. And what is centrosome actually? Regions where microtubules polymerize. Okay, this is going to be the best answer from here. I read it as centromere. Sorry. Okay, in the cell cycle, mitosis occurs between dash and dash. Yes. So tell me, guys, what do you think? Very nice. Darshanita is right. Kanaka is right. Yes. Vaishnavi is right. Waiting for Zui. No. Waiting for Arshia. Arshia, quickly. See, guys, in cell cycle, mitosis is going to occur between what? You are going to have, see, mitosis we said out over here, we are going to have G1, we have your S phase, we have G2 phase. So where is it? In between G1 and G2. That way. Okay, so answer is that. Okay. Actually, better would have been that, if they would have stated G2 and G1, then it would have, not created any confusion. Anyways, cellular structures which disappear during late prophase and reappear during anaphase. Yes, my question goes to Kanaka. Kanaka, tell me. Very nice, very nice. It is going to be nuclear membrane and nucleolus. Very nice. Okay, the best stage to observe shape, size, and number of chromosomes. Yes, Ruby, very nice. Yes, good, everyone. It is metaphase. Okay, next thing. Okay, next question goes to Tanishka. A picture of a dividing Drosophila cell through a microscope show that the cell contain 8 chromosomes, each consisting of 2 chromatids. The picture must have been taken during. Guys, think about this. They are saying that, oh, they are looking into a Drosophila cell and they happen to find out 8 chromosomes. But these chromosomes has two chromatids each. What is this? Yes, it is going to be mitotic metaphase. Very nice. Okay, which of the following phase of mitosis is essentially the reverse of prophase in terms of nuclear change? Very simple. Yes. Quickly, guys. Yes, it is going to be telophase. Okay, which of the following is not a function of mitotic cell division in animals? Which of the following is not a function of mitotic cell division in animals? One minute, guys. Which of the following is not a function of mitotic cell division? So just see what is it, guys? Gamete formation. Okay, earlier everybody was giving me some different answers. Anyways, okay, to form 16 cells from to form 16 cells, how many times mitosis is supposed to occur? To form 16 cells. Yes. Very nice. Zui has given me the right answer. Yes, everybody is giving me the right answer. Yes, it is actually this option is actually B. Okay, it is going to be 15 times. Okay, so actually how it is going to be, 15 times. Why mitosis has to occur? See, one cell, it is going to give rise to two cells. This is going to be your first mitosis. Okay, but see, division is taking place out over here also, right? So they are also dividing. So to form 16 cells from one cell, guys, always remember n is equal to number of cells minus 1. That many divisions, okay, that many mitoses you are going to require. Actually, the answer is 15. Okay, But now there are two ways in this thing, like, how this can be done. Okay, actually, see, this is like, how many cell division, how many mitosis, how many times mitosis is going to occur. This is the thing. Okay, but guys, then there is another question: how many times cell division has to occur? If they ask you, like, how many times or how many generations are required, then you will see that generation number is going to be 2 raised to n. So for example, let's understand this thing. See, in order to form 4 cells, how many generations of mitosis I have required? See, I have required 1, I have required 2. 2 generations of mitosis. Okay, but in turn, how many mitosis, like cell divisions I have required? Cell divisions are going to be 4 minus 1, that is 3 cell divisions. It is that way. Okay, so for the time being, guys, you just need to keep it in mind that when we talk about the number of cell divisions or the number of cells, it is always going to, the number of mitosis, it is always going to be number of cells minus 1. Okay, keep this thing in mind. Okay, the members of a homologous pair of chromosomes, guys, yes, it is right, are identical in size and shape and appearance. Right. Okay, next, the 4 haploid nuclei found at the end of mitosis differ from one another in their exact genetic combination composition. Some of this difference is a result of. Why is this happening, guys? Can anybody tell me quickly? The difference is going to be because of, yes, crossing over during prophase 1. Very nice. Okay, 4 different steps that occur during meiosis are given in the following list. Okay, we have to pair this thing up. So guys, just look at the steps and let's try to see who is going to be first, who's going to be second, who's going to be third and fourth. Yes. Okay, so complete separation of chromatids, pairing of homologous chromosomes, lining up of paired chromosomes on equator, crossing over between chromatids. Yes. So guys, who's going to be the first one? Like, just tell me who of them is going to be first one? 1, 2, 3, 4. 1, 2, 3, 4, guys. Yes, this is going to be the first one. Then next one is going to be the next one. Next one is going to be, yes, it is crossing over. Sorry, this is going to be crossing over. Then third, it is going to be, no, the complete separation of chromatids and chromosomes on the equator. What is it? Yes, it is going to be this. And fourth one is this. Let's try to see what is the answer, guys. Answer is 2 3 no, 2 4 2 4 3 1. Yes, this one. Okay, let's try to see the next one, guys. 2 more questions and we'll be done. Okay, the DNA content of individual cells and the number of cells in each phase of cell cycle can be determined using flow cytometry. Which of the following combination of phase of a cell cycle and its corresponding DNA content can be considered normal? Okay, diploid cells. Array, what is this? Oh, there is some error. Oh no. Uh, oh no. No, 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 guys. We can't solve this thing. There is an error out over here. Janita. Last one. Okay. Sarah would like to film the movement of chromosomes during cell division. Her best choice for microscope would be, she is she is trying to film the movement of chromosomes. Okay, so what would her best choice would be? Light microscope because of its resolving power, transmission electron microscope, scanning electron microscope, light microscope because the specimen is alive. What do you guys think? Light microscope meaning your compound microscope. Yes, Akanksha has given me the right answer. Vaishnavi is also right. Now, guys, the right answer is light microscope. Because remember, not because we are going to go for its resolving power. Light microscope doesn't have a great resolving power. Okay, but why will we go? Because the specimen is alive. Otherwise, what in transmission microscope, the specimen has to be killed. Scanning electron microscope, the specimen has to be killed. That is all. Okay, everybody all right with this thing, guys? Sure, no problem. Okay, so guys, once again, we'll stop over here for the day. Next class, when we come back, guys, once again, I will get the other this thing, uh, the other files. Basically, I don't know where those files have gone now. I will go get those files, and then we'll start once again solving some more question and answers. But in that case, we'll do only selective ones, like typical of what is generally asked in your exams. Yes, I will be sending this on to the group. Okay, sure. So guys, we'll stop over here for the day, and I'll see you in the next class. Till then, take care. Bye bye, everybody. Bye bye. Bye.